I'm very really excited to be here to talk with you guys about some of the work that Matt and I have been doing here at USU for my PhD. I've been working with the Soil Health Partnership out of the Midwest, and I'll talk about some of their work. And so the basic objectives that I'll cover today is some of the characteristics and definitions of the soil health tests and assessments that are in use around the country today. And then I'll discuss a little bit of the research that I've done for my dissertation about the performance of these soil health assessments and what we can learn from them about soils and also about productivity. So just to start off with, I know since we're here to talk about soil health, most of us are all very familiar with the concept of soil health is essentially the ability of the soil to support and provide for human and natural needs without degrading the environment. And so here I've listed some of the characteristics of a healthy soil system and also the functions of the soil there in that diagram on the right. And I think that it's really important to come back to those functions, to refer to our analysis of soil health as an indication of what is the soil doing for us? What are we trying to gain from the soil, whether it's going to be some kind of filtering and buffering of chemicals in the soil, or if it's just nutrient cycling, or just supporting plant growth as a medium for growth in good aggregate stability, for example. Really, I think what all of these characteristics boil down to is having a healthy soil system means that it's going to be resilient to degradation. It's that the soil is going to be able to support the growth of the crop, and it's going to provide a habitat for the microbes and the insects and the animals that are going to live there that are going to be beneficial to our crops as well. So essentially, soil health is a combination of the biological, chemical, and physical properties of the soil. And it's this combination, soil health is an emerging property of these characteristics that are diverse, connected, interdependent, and adaptive. So when you take a step back and look at soil health in this context, soil health really becomes this concept. And what soil health means here in Utah is, it's going to be specific in this context, and it's going to be a little bit different from what soil health is out in the Midwest. And I'll get into a little bit of that and some examples later on. So some of the major indicators of soil health, as I talk about soil health as being this concept, soil health itself isn't something that we can particularly measure, but we can measure things in the soil that can indicate certain attributes of its functionality. So we can measure the organic matter, we can measure the organic nitrogen and carbon as an indication of the nutrient cycling functionality. So each of these things that are indicating soil health is going to be something that we get a lab value for, and then hopefully we can try and understand something about the total system from that. So back in the mid-90s and early 2000s, soil health researchers started to develop soil health assessments and soil health tests, trying to take these indicators of soil health and trying to put them into kind of a score, something that can convey some information about what is going on in that soil system. kind of summarizing all of that information from each of those lab values. So first you start out with a minimum data set of the physical, chemical, and biological indicators. And that previous slide that I just showed was a very small list. I've seen researchers studying up to 80 different indicators of soil health. So the first thing that you have to do is boil that data set down. You can't take 80 different indicators and try and make sense of all that at the same time. So you really want to summarize things down and condense that information down as best you can. And then the interpretation of the indicators, they've developed scoring functions. The midpoint optimum, more is better, and then less is better. So the midpoint optimum would be an example of, say, phosphorus, where your soil health indicator value, going along the x-axis there, if you have a kind of an average phosphorus value, a good phosphorus value in your soil, then your crop productivity will be better. Another example of that would be with soil pH. If you have a low pH or a high pH, your crop productivity isn't going to be as well. So the score then that we give to pH is going to be high in that midpoint range, and so forth with the more is better and with the less is better as well. An example of the less is better would be sodium content in our soils, and that's especially something relevant here in Utah, as Neil pointed out earlier, where our sodium content that would return a low sodium content lab value would get a higher soil health score from that. So 3 major soil health tests have emerged over the last 2 decades. First, the Soil Management Assessment Framework, or the SMAF, was developed in the mid-2000s by Doug Carlin in Iowa. And that one is mainly used by researchers. It uses algorithms and logic statements to score those functions, to score those indicators in that kind of a function there. The Comprehensive Assessment for Soil Health, developed by the Cornell University Soil Health Lab, uses a statistical function, and it was initially based on the SMAF. And that's available for commercial use. And then the soil health tool developed by Rick Haney out of Temple, Texas at USDA ARS. That's also commercially available, and many of you are familiar with that. And I'll get into that in a little bit more detail as well. So the Comprehensive Assessment for Soil Health looks something like this. It has a very robust suite of soil health indicators that it measures for. Soil texture, it gives you that. Available water capacity. Penetration resistance is one of these novel things that you use a tool that can measure the pounds per square inch of pressure that the soil would resist as you push down and this tool. It's actually a pretty neat tool that you can use to help gauge how much soil compaction that you have in your field. The cache also uses the soil respiration on a 96-hour test, whereas the Haney test uses only a 24-hour, just for your reference there. It also has a root pathogen pressure test that they actually take a large sample of your soil, and then they plant whatever crop it is that you're going to be growing in there, and then they'll test that for root pathogens. And they have this available in different levels of packages. So you can do the basic or the standard or the advanced, and each of those have a different cost associated with them, and I'll show a graph of that here in a few minutes. There on the right is a a representation of the kind of report that you would get if you were to send one of your samples in there to the Cornell Soil Health Lab. They provide the lab value that they tested for, and then they give you the rating. And then on the right, it shows you the kind of constraint that you may be facing that would be causing you to have a low soil health score. And that part of this soil health test is, I think, really the most important aspect of it, is that it's going to show you, for example, in red there, the soil protein index has a low rating. So it's going to tell you that your organic matter quality and organic nitrogen storage is constrained in some way. So they're then going to provide you with some recommendations of how to remedy that. So the idea is that you send your sample into them, and then they can provide you with an assessment of some ways to overcome some constraints that would be restricting your soil health. So the CASH assessment is based on basic statistics, the average and the standard deviation within the calibration data set that they're using. And in 2016 and 2017, they released updated statistics for this, that their scores are based on regional values, because they found that from one region to the next, the average soil organic matter is going to be different. That's really no surprise, but that has an effect on the scoring. The cumulative normal distribution, it's based on the lab value. So you think about the normal bell curve. This is just a cumulative version of that. So your low value is going to receive a score of 0, and your highest value is going to receive a score of 100. Your average indicator value would receive a score of 50. So if you go from the light blue region in the far northeast to the ice states in the dark blue there, your average soil organic matter or active carbon or even the penetration resistance may change. So your score is going to be different. And I'll show some more examples of that from the Midwest in just a few minutes. All right, now so for the Haney soil health test, that score is a multiplicative index. So they take the 24-hour CO2 burst or Sol-Vita test multiplied by the value of wet extractable organic carbon and wet extractable organic nitrogen. So these are the values of carbon and nitrogen that are most accessible to the microbes and to the plants in the soil. And so this Haney score index is really showing us what kind of biological and chemical activity is taking place in the soil. On the right is an example report for the Haney test from Ward Laboratories. And they're going to give you a whole lot of response variables that they test for. But the Haney test itself is really intended to be managed, is to help you manage your soil fertility. And there's a lot of really great work that Matt has done on this that I can get you the references for if you'd like to learn more about that. But within these three different components, the Sol-Vita, and the organic nitrogen and carbon, we wanted to find out which one of these components represents most of the variation in your soil health score. And this was, again, work that Matt did, that he found that the Sol-Vita test represented around 80 plus % of the total variation in your soil health score. So I know that the NRCS cost shares the full Haney test, But if you have a grower that may not want to pay for the full cost of the Haney test, then instead of doing everything, you could just do the CO2 burst and get most of that same information. So that really has implications in managing, your costs for introduction into these soil health tests. And here's an example of some of the approximate costs. On the left are individual soil tests. So if you wanted to go out and do a la carte testing, you could do that, and the cost would be much less versus the cash basic or the standard or the Haney test. Now, at a cost for the cash standard at around $110 per sample, it's going to get cost prohibitive quite quickly. So if you're going to ask me what should you start with, if you're going to recommend to a farmer what you should start with, I'd say basic organic matter. It's something that they're probably already doing. But then I would also say to start with soil respiration, getting an understanding of the biological activity that's going on in the soil. And then I would also say the wet aggregate stability to give you a good understanding of how well your soil is holding together. Those three things I would say would be a great introductory soil health assessment. From there, you could just get into the basic packages and so forth. Now, how well do these tests relate? Since they're testing similar things, we wanted to run some regression analyses, and we found that the correlations were dependent on the soil texture. Now, the cash overall score. So for the cash index score, you take each of these indicators and then just average them together to get an overall soil health score for your field, as opposed to the Haney test, which is just a multiplicative calculation, like I showed. Anyways, so the point here is that these relationships really aren't very strong. So maybe that is suggesting that if you wanted to go out and do these tests separately, then it may be giving you some different information. And this is ongoing research that we're still trying to learn a little bit more about. Now, how much variation can we expect in these soil health tests? For the cash index score, the average temporal coefficient of variation was very similar to the Haney test. So from year over year, we found it's about 20% CV. But spatial coefficient of variation is a little bit different. And I think the Haney has the higher coefficient of variation because it's based more heavily on those biological characteristics. And we know that soil biology changes from 2 feet away. So I think that's why we're seeing the Haney test is going to have higher spatial variation. Now on to the Midwest case study. So the Soil Health Partnership is a grower initiative started by the National Corn Growers Association. And it was started in 2014 with the primary mission to help farmers start to implement soil health practices and then to gather data so that they can make data-informed decisions on how to maximize their soil health in economic ways. Because we can't go out there and say, okay, you should convert to X practice with this kind of crop rotation if it's not going to help them in their management and with their economics. So the Soil Health Partnership really is focusing on how to help the farmers adopt these practices, but then also make it work for them at an individual level. This map shows a little bit of the distribution of the sites that they have across the country. It's focused mainly in the Midwest right now. There are over 100 full partnership sites. And the experimental layout has 4 replications of a soil health treatment versus the farmer's conventional. So this could be a cover crop or a reduced tillage or a combination of those two. And we have this really robust, very large data set now with site management and site environment characteristics and factors. And this is the data set that I'm analyzing for my PhD dissertation. So first, what can we learn about soils from these soil health tests? First, I wanted to look at what do these scores look like and some of the basic indicators that we have. So first up is soil organic matter. So the scores here are, I have calculated myself. So if you were to send in the scores, then you would get, if you were to send in your sample, you would get a score based on the Cornell data set and what they have. What I've done here is I've taken the Soil Health Partnership data set and I've calculated the CASH score based on our own data set. And a lot of people have said that if you were to take a soil sample here in Utah and send it in to the Northwest and have them analyze it, the soil health score that you would get would be way off. And you'd be right, because their data set for this region isn't as robust as what they have for those regions back east. So for the analysis that we've done with the Soil Health Partnership is that we wanted to go ahead and see what exactly it was with the region that we're dealing with. So it's interesting to look, and I'll just skip ahead here, to show in summary how the soil texture is affecting the soil health score overall. Because the average soil organic matter or water holding capacity or wet aggregate stability, the average is changing with the soil texture. And if we were to put these same graphs up here by region, we would see that same thing, that these soil health scores are going to change by region and by soil texture. The one thing that I do want to back up here is to show you in the wet aggregate stability, the medium soil texture had the lowest average value. And on the bottom right, this is a regression showing the intensity of tillage and that effect on the wet aggregate stability. So from 0 to 1, 2, and 3 is increasing in tillage intensity from no-till, strip till, vertical till, and then a conventional full disc tillage. So even though the regression relationship is very weak, it's still a statistically significant relationship. And this is something that we have to dig into more. That it's interesting to me that the aggregate stability, that the average aggregate stability is higher in a coarse soil than it is in a medium textured soil. I think this is really interesting, and this regression relationship shows a little bit about that. All right, so these show some of the relationships between, on the left, the percent clay and the soil respiration in the Haney test, and percent organic matter. Again, these relationships are very weak, but I still think that there is something important that we can learn here about this, that if your organic matter increases, then your biological activity is going to increase overall. And that's basically what we can learn from that Haney test. All right, what can we learn from crop productivity? So these graphs are examples of regression relationships at one site over 2 years. This is the corn grain yield and a soil health indicator relationship. And I've boxed out the indicators and the seasons that had a significant relationship. And so I dug into this a little bit and tried to find out why did year 4 yield so much more than yield 2. And it was really interesting that it really came down to just the fact that this site had been in the partnership for 4 years, and so there was just this trend toward a higher yield. Year 2 actually had higher rainfall than year 4 did, by a few inches actually. And I thought that was very interesting. And this is a trend that we've seen across the whole partnership, is that as sites have been in the partnership, their soil health scores have increased and their yield has increased. So that's also something that we're digging into a little bit more as well. This bar chart is showing a summary of 107 site years, so similar regression relationships. And I just wanted to show how many site years had a significant correlation. And you say, okay, well, if there are only 7 or 8 site years out of 107 that had a significant correlation for corn yield and the cash score, when we're saying, okay, go out and take a soil health sample, have your soil tested for your soil health assessment, get a score, and then make a management decision based on that. But you're saying that the score and yield are only correlated only less than 10% of the time. I think that in this case we just have to take a step back and say that in 1 year we could have a statistical relationship, and in another year we may not. But over time, see, that's what really is the concern, is that over time, if we're seeing a relationship between building organic matter and higher yields. So if your farmer gets a report back and they say that this score really didn't match up with the yield that I had this season, say that's okay. You're not always going to have a direct statistical correlation. What matters is over the next 5 seasons, 10 seasons, even 20 seasons, as you measure your soil health, are you going to be having a better outcome in your soil functionality? See, that's really the key here. So just to summarize really quickly, I think that it kind of, and I'm just about out of time here. In soil health, we're trying to build a house. We can't build a house in 1 day, or in this context, we can't build our soil health in just 1 season. So when we go out and help our growers to understand why it's important to measure soil health, you say that, well, you're trying to build a home for the soil microbes. You're trying to build some structure so that your plants have somewhere to grow and to get their water. So I think just taking that step back and looking at it in that context, that's really what's important in selling that soil health message. And I just want to thank my co-authors and collaborators, and there's about a minute left. I can take any questions if you have any questions.